All right, ladies and gentlemen, welcome back. We're going to continue our discussion of the endocrine system today by talking about the actual pathways in which these hormones travel throughout your body to ultimately cause some sort of targeted response. So, when we look at the secretion of these hormones, where do they happen? How do they get to their target cell? And then what are those actual responses that are being created is really the pathway that we're talking about. So let's start off just talking some simple hormone pathways. So when we discuss a hormone pathway, we're looking at the release of that hormone from its endocrine cell, it traveling through typically the bloodstream, and then ultimately that hormone gets to its target cell, interacts with its specifically coordinated shaped receptor, and then causes some sort of targeted response in that cell itself. So, if we look at a particular example of a simple hormonal pathway, we might be able to look at something like this, where we take your stomach, you've just eaten, you're digesting your food, and as that food digestion continues to occur, the acidic contents of your stomach get released into your duodenum, which is right here, which is the first bend into your intestines. And when we cause that to happen, we're actually causing the pH in your duodenum to drop. This tends to be a little bit more on the basic side. This is considerably more acidic. So as I empty that contents into that duodenum, your pH in your duodenum drops. That's our initial stimulus. What that ends up doing then is causing endocrine cells that exist in the lining of that duodenum to secrete a hormone called secretin. That secretin then gets released into the bloodstream and travels to the pancreas, which is right here. Okay, so the target is the pancreas itself. What does that end up doing? Well, that pancreas then releases a bicarbonate molecule, which is the ultimate response. What does that do? Well, this all acts as a negative feedback. So as that pH is dropping in the duodenum, we're telling your pancreas that this is happening. The pancreas then responds by secreting bicarbonate, which pulls the pH back to a more basic level, and in fact increases or heightens that pH. And that will continue to happen until we retain the desired pH in the duodenum, we retain homeostasis. We can also look at, rather than it just being simple hormonal, we can talk about simple neuroendocrine pathways. And this particular example here is one that is a positive feedback instead. So here the stimulus is received by a sensory neuron, which is then stimulating a neurosecretory cell to release neurohormones instead of a traditional hormone. That neurosecretory cell releases that neurohormone, which then enters the blood and travels to its target. So if we look at the example here, all right, we've got some suckling. This is a breastfeeding, so the suckling at the breast, that's the stimulus. That then elicits a sensory neuron into your hypothalamus, a female's hypothalamus, to release oxytocin, which is a neurohormone, which travels to the blood, goes down to the smooth muscle of the breasts, and allows the milk to get released. And so long as that suckling happens, this continues to build up a continuous loop that continues to produce the milk, release the milk, until the suckling action stops. So, really, when we talk about looking at these pathways, we're looking at feedback regulation. These hormones are assembled into regulatory pathways, and the whole purpose of these pathways is to ultimately ensure that the body is capable of maintaining homeostasis. That communication in maintaining that homeostasis is really what allows the body to do its job. Any discoordination of that communication system, that throwing off of that homeostasis, ultimately leads to systems degrading, not doing their assigned jobs, and as a result, the organism overall ends up suffering, which can ultimately lead to death. Now, that hormonal control is only part of that feedback. Most of the time, you're looking at both a coordination between the endocrine and the nervous systems to maintain those internal stable conditions. Interesting thing is that all hormones, regardless of whether they control or work in a positive or negative feedback mechanism, is that they all are ultimately there to maintain that homeostasis by altering some sort of physiological activity of the target cell itself. So when we look at feedback regulation and how those hormonal system controls work, they are all relatively consistent. So there is ultimately really 5 major pieces to any control system, regardless of whether it's positive or negative. So first and foremost, there has to be some sort of stimulus. What is the factor that's being regulated? You then have a receptor to detect what that change is. So, what is the change to that typical receptor that we're monitoring? These receptors are monitoring both the environment and then ultimately the response to that stimulus. You have then input of information. So once we've detected the stimulus, decided what it was, decided what it's doing at the local level, that information then travels along your afferent pathways to your control center, which in us is our brain. And then it's the job of that control center to decide what the appropriate response to that stimulus is and how to work with it. That information then comes back through the output. So it's sent back through the central control system, travel down the afferent pathways to create some sort of effector, which ultimately becomes your response at the end to balance out that original stimulus, or to potentially push it forward, depending on whether we're talking positive or negative control systems at work here. So this is the basics of what we're looking at. When we talk about control systems, usually we're talking about some sort of variable that becomes imbalanced. That's the stimulus. We detect that via our receptor, which sends the information to the brain, puts out an effect, and hopefully pulls everything back into balance. Most feedback mechanisms that kids think about are negative control mechanisms or negative feedback loops. These are the ones where it really is like the seesaw mechanism, where I've caused an imbalance and I'm going to do the opposite to balance it back out. So the majority of our homeostatic control mechanisms work by negative feedback. Think about things like your body temperature. You're getting too warm, so what does your body do? Produces sweat. That evaporative cooling then brings your body temperature back to homeostasis. Blood pressure works this way, your blood sugar regulation, red blood cell production. There's any number of feedback mechanisms that you think about, more than likely are negative feedback. So we're pushing you in one direction, and then your control system brings you back the opposite direction to keep you at homeostasis. Positive feedback, on the other hand, actually reinforces the stimulus and causes an even greater response. So rather than doing the opposite, we're continuing to reinforce whatever that stimulus was in the first place. Things like childbirth or blood clotting, lactation, these are all examples of positive feedback mechanisms where we continue to enhance that effect until we get some sort of desired response. Once that response is met, then that loop gets broken, and we suppress the loop and everything goes back to normal.
Hi, it's Mr. Anderson, and welcome to Biology Essentials video number 18. This is on positive and negative feedback loops. If you've ever driven, you've probably seen a sign like this. This is a guilt sign. So they put the speed limit up here, and then they show how fast you're going down at the bottom. But you constantly get feedback as far as your speed goes. So you will see that you're going a little fast, and so you'll slow down. But then you'll realize you're slowing down way too much, and so then you'll speed up. And if it works out well, you'll hit that speed limit. or that target set point. Now, what I'm just showing you here is what's called a negative feedback loop. In other words, you'll dance around that point. You'll speed up, you'll slow down, and eventually you hit that point. So a negative feedback loop brings you closer to the target set point. A positive feedback loop you would experience if you were to see this sign as a challenge. So you see you're going 30 miles an hour, so you realize I could go 39 or maybe 51. I could eventually get a ticket or break the sign. So that's a positive feedback loop. It's when you're amplifying and moving away from that target set point. So in this podcast, I'm going to start by talking about homeostasis, and that's our internal environment. And to maintain a stable internal environment, we use feedback loops. With each of those, you have a target set point. In a negative feedback loop, you're going to move above or below that, but try to stay as stabilized, and so you're as close to that as you possibly can be. Example I'll give you is temperature regulation in mammals. In a positive feedback loop, you move away from that target set point, so you amplify that. Example I'll give you is fruit ripening, why all the apples on a tree seem to ripen at the same exact time. Now, with a feedback loop, you can always have mistakes. You can always have alterations in that feedback loop, and that can lead to tragedy. Example would be diabetes is a problem in creating insulin or sensing that insulin. And so that's just a problem in a feedback loop, and it can lead to really bad things. And so let's start with homeostasis. Homeostasis, if we were to define what it is, it's an internal stable environment. And so if you live in a pond, if you're a paramecium like this and you live in a pond, you let a lot of your environment just go, and you maintain that environment by just maintaining the same as the pond around you. So you have this external pond, and whatever the temperature of that is, that's the temperature of the paramecium. Now, they'll regulate a few things like water concentration using contractile vacuoles, but mostly they have a pond. Now, as we move out of the pond, or as we become more sophisticated, we kind of bring that pond with us. That internal pond now is what homeostasis is. And so this is a hairless cat. Hairless cat is going to maintain its body temperature, and so it does that through feedback loops. It's going to maintain its blood glucose level. It does that through feedback loops. It's going to maintain the osmolarity of its blood. It does that through feedback loops. Now, hairless cat doesn't have hair, and so it's actually hard to maintain its internal body temperature, and so you don't want to let them go outside on their own. They'll lots of times put a coat on so they can maintain that. Now, there are kind of two life strategies in this picture. It'll take you a second to figure out what it is. This is a snake eating a rat, and it's taken with a thermal image camera. And so the snake itself is what's called an ectotherm, and what that means is that their internal temperature is going to be the same as their external temperature. So the snake is about the same as, whatever the counter that it's sitting on. But the mouse is going to be an endotherm, and so that is going to keep an internal temperature that's going to be constant. Now, there's some advantages. If you keep an internal constant temperature, then all the metabolism will work at the same exact rate. The problem with that is that the rat right here is probably going to eat a lot more than that snake just to maintain that body temperature. So let's see how that works in a mammal. And so in humans, we use a negative feedback loop to maintain our constant body temperature. And so our constant body temperature is around 37 degrees, or about 98.6 degrees Celsius. And so we use a negative feedback loop to maintain that. So let's say we put a thermometer in this kid's mouth, and the temperature all of a sudden increases. Now, you actually sense that. The area where we sense that is on the roof of our mouth, in an extension of the brain called the hypothalamus. And so we're going to sense that temperature change. And so what's the first thing that your body does when you start to get too hot is that you're going to sweat. And so what does that do? Well, as these water molecules evaporate, that's going to create evaporative cooling. In other words, they're going to carry a little bit of heat with them. You'll also start to vasodilate. In other words, the blood is going to be carried towards the surface of the skin. And so then just through convection, we're going to start to lose more of that heat to our environment. Now, another thing that would happen if we had fur, which we don't really have, is that fur is going to lay really flat. And the reason why is that then we can have more of that heat being lost through convection. So what does that do to our temperature? Well, it's going to drop. but it might drop a little bit too far. So then we're going to turn those things off. We're going to quit sweating, and we're going to quit vasodilating. But now we're too cold. So what do we do? How do we keep ourselves warm? Well, we're going to start to maybe get goosebumps, and these are kind of crazy goosebumps. But we're going to get goosebumps right here. And what goosebumps do is, if we had hair, it actually stand the hair up on end. But it also kind of pulls your skin in. It's like taking a coat and kind of pulling it in, and it's conserving that heat. We're also going to vasoconstrict. In other words, we're going to shut off those capillary shunts, and we're going to hold that blood in towards the inside of our body to maintain that. And as a result of that, we're going to have less convection, and our body is going to increase until we hit that target set point. And so right now, in fact, throughout the whole day, your body is just regulating your body temperature, and it's doing that through a negative feedback loop, again, trying to maintain that target set point. What happens if we want to go in the opposite direction? Well, let's say we have this. So we've got a fruit on a tree, and we call that the target set point. But let's say we want to move from fruit on a tree to fruit that is ripe. Now, why do trees make ripe fruit? I could talk about that for a long time. What they're essentially doing is making an attractant. So birds and humans are going to come and eat the apple and spread the seed somewhere else. But how do they maintain that? Well, they communicate, and it's kind of hard to understand how a plant could communicate, but they communicate through a plant hormone called ethylene. Ethylene is just C2H4, and it's given off by ripe fruit. In other words, this gas is going to be given off by ripe fruit. It's going to be picked up by apples next to it, and then it's going to cause them to ripen as well. And so if you take one nasty, over-ripened apple and put it in a barrel of apples that aren't ripe at all, they will all ripen as a result of that ethylene. And so one bad apple can really spoil the whole lot. But how is this an example of positive feedback? What goes on is that first apple will become ripe, and so it's going to start giving off ethylene. And that ethylene is going to be picked up by apples right next to it, and they're going to create more ethylene, which is going to create more ethylene, which is going to create more ethylene. And so through this positive feedback loop or amplification, all of a sudden all the fruit on the tree are ripe at the same exact time. Another famous example of positive feedback loop would be in childbirth. The pressure of the head on the cervix of the mother actually causes contractions, which pushes more pressure on the cervix, which causes more contractions, and so eventually the baby is born. And so we see positive feedbacks when we want something to happen really, really quickly. It's not something that we maintain for a long period of time. So what happens when something goes wrong, or what happens when there's a mistake? A great example of a feedback loop in us, so this is a negative feedback loop, would be blood glucose levels. And so the blood glucose level is the amount of glucose that's moving around the blood in your body. Where else could it be? That glucose could also be taken in by the cells, so they can do respiration. We can get ATP from it. Or we could also store it in glycogen, which is mostly going to be found in the liver. And so we use two different hormones, insulin and glucagon, to do that. And so this is your pancreas. Pancreas is going to, its major job is to empty digestive enzymes into the small intestine when there's food there, and so we can break it. But they break it down or absorb that, digest and absorb it. But they also have a dual purpose, and one is to regulate the blood glucose level. And so they have two types of cells in here. They have beta cells and alpha cells. And so the beta cells and the alpha cells are they're just sensing the blood glucose level. And so this is an example of a beta cell right here. And so this is our glucose transport. It takes glucose in. This would be cellular respiration here. And so if we have a lot of blood glucose outside the cell, in a beta cell in the pancreas, that's going to trigger an influx of calcium, but more importantly, it's going to increase the amount of insulin that it's giving off. And so what that means is that when the blood glucose level is high, insulin, which I'll represent with this red kind of a dot, is going to be secreted from the pancreas. And that's going to move throughout your whole body. And it's going to trigger cells in your body to take in that blood glucose. And it's also going to tell your liver to store that as glycogen. And so when your blood glucose level goes too high, insulin is secreted, and that's going to cause your blood glucose level to go down. What happens when your blood glucose goes down, so dangerously down? Well, then your body is going to quit producing that insulin and it's going to start producing glucagon. Glucagon is going to be created by the alpha cells in the pancreas. And so now when the blood glucose level goes too far down, glucagon is added, and that's going to increase the blood glucose level. In other words, it's going to free up glucose from glycogen in your liver. It's going to increase the blood glucose so you can actually use that throughout your body. Now let's actually see how this works throughout the day. So this would be in a typical person. What we've got here is the glucose levels in red, and so the glucose is going to increase, but it's increasing 3 times a day, and that's because you have breakfast, lunch, and dinner. But if you look at this dance, is like right next to it. This is this dance between the blood glucose level, which is red. As it goes up, the insulin level is going to increase as well. What does that insulin do? It causes the blood glucose to drop, and then we're going to increase insulin again. And so we get this cycle throughout the day of an increase in blood glucose, increase in insulin, and then we keep that at that level. And so if you look at it, our blood glucose levels throughout the day are going to maintain a fairly static kind of a position. Now, if we were to eat smaller meals throughout the day, that would probably help us keep that together. But what happens if we have a mistake? What if we have an alteration in this feedback loop? Let's say, for example, that you are type 1 diabetic. Type 1 diabetic, the problem with type 1 diabetics is that they have beta cells that don't work. So remember, the beta cells inside your pancreas are secreting that insulin. But if you have type 1 diabetes, or sometimes called that child onset diabetes, you have destroyed these cells. It's usually, there's a genetic component to it, but it's mostly an autoimmune disease where you destroy the beta cells. Well, what happens if you destroy the beta cells? Now we're going to have a feedback loop where the blood glucose comes up. There's no insulin to secrete, and so blood glucose keeps going up and going up and going up. And so your cells aren't going to take in that blood glucose, and so we have some nasty stuff that comes as a result of that. And so here's some things that happen. Increases the blood pressure, can eventually affect the eyes. You have nausea, vomiting. A lot of the ones in blue here are for type 1 diabetics, but it eventually can lead to putting you in a coma or actually death. So if you're a type 1 diabetic, that's just a mistake in the beta cells in the pancreas, so we can't make insulin. If you're type 2 diabetic, that essentially means that you've had too much glucose throughout your whole life. It usually is tied to lack of exercise, obesity, things like that. Type 2 diabetics, the cells in your body just stop recognizing that insulin, and as a result, they quit taking it in. And so how could we solve that problem knowing what you now know? Well, if you were to get insulin shots, like an insulin shot right here, or an insulin shot right here, or an insulin shot right here, and insulin, I think, was first synthesized or created in the 1920s, but now we make it through molecular biology. But if you could get insulin shots throughout the day, then you can regulate that blood glucose. Or now they use a insulin pump, which is going to administer different amounts of insulin throughout the day, so it works almost as a feedback loop. Now, this is a map of where diabetes is on our planet. About 3% of the people in the U.S. have, or worldwide, have diabetes, but about 90 to 95% of that is not a mistake in the beta cells. It's actually tied to our diet, type 2 diabetes. And this is a chronic disease. Once you get diabetes, you have it for the rest of your life. And if you look here, where are we seeing the greatest incidence of diabetes? It's just tied to our diet. And so we're eating a high fat diet, and as a result of that, the diabetes, a high sugar diet, corn syrup diet, and as a result of that, we're getting huge diabetic increases. But the whole thing is tied to a mistake in a feedback loop. And so that's feedback loops, positive and feedback, positive and negative, and so I hope that's helpful.
So when we sit back and really analyze how your nervous and endocrine systems work together, there really is a lot of coordination in these feedback loops between this neuroendocrine and endocrine signaling. So those endocrine pathways are subject to a lot of regulation by your nervous system, including your brain, because we're sending those signals to your brain to be interpreted, and then the response back may be endocrine related. So they generally act in coordination to control all sorts of things, including reproduction and development. And although overarchingly we're sitting here focusing on what this looks like in the human body, let's take a step back real quick and just remember that this stuff is going on in other organisms as well. In fact, it does a lot of control of reproduction and development in all sorts of organisms. One just particular example are the larvae of butterflies and moths. They get these signals that directly impact their ability to go from sort of that caterpillar stage into that adult butterfly. So the coordination of their nervous and endocrine systems directs that sort of molting, and it ultimately does originate in their brain. So, in a lot of insects, and this is just one example, that molting and development is controlled by a combination of hormones that are ultimately regulated by the brain itself. So, this is just one example that your textbook goes through, and a couple of hormones that end up working in conjunction here. The PTTH is a brain hormone that actually stimulates the release from one of their glands, these ectosteroids, more specifically from their parathyroid gland. They also have a coordination going on with a juvenile hormone or GAH that promotes the retention of their larval characteristics so long as that's in high concentration. Along with this ectosteroid, they'll sort of stay in these early larval stages. And then when that low GH comes into play, but that ectosteroid is still being produced, that's what then causes the production of the pupa and then ultimately the adult. So this ectosteroid is there at all times, but then it's coordinated based on how much of that JH is in circulation. So long as there's a lot of it, it will stay in these larval stages. And once that drops off, we get to the pupa. Once it's gone completely, then we move over to that adult stage. So when we look at what this does look like in humans, we've got a lot of coordination of neuroendocrine, endocrine signaling. More specifically in us, we look at your brain and there's a handful of endocrine glands there that are particularly important in regulating all sorts of releases of hormones from some of the other glands in your body. So we look here, all right, just below, right by your brainstem is your pituitary gland, and that pituitary gland is broken into two pieces. You've got your posterior and your anterior, then your hypothalamus is sitting right here. So that hypothalamus's job is to receive information from your nervous system and initiate all of these endocrine responses. So when we look at those feedback loops, information may be coming into your hypothalamus that then will direct your pituitary glands to produce certain hormones, secrete certain hormones that will then have cascading effects throughout your body. Your pituitary gland stores and secretes hormones that are made specifically in your hypothalamus, and then your anterior pituitary gland makes and releases hormones under the regulation of that hypothalamus.